at Imperial over the last 6 to 9 months, and also speaks to much of what you've heard today. And what it really addresses is on the left are, I think, the important questions about that need to be asked and answered as we implement whole genome sequencing. And so reading down that list on the left, and much of this takes account of what you've heard today, we have the question of how best to harness the power of whole genome sequencing in order to maximize public health impact, to rethink contact investigations and inform and develop the next generation workforce for TB control, as you heard about from Philip Monk and Grace Smith, individualizing patient management and precision medicine, as you heard about from Sarah Fortune just now, and also Derek Crook, correlating genotype with phenotype and insights that can bring into pathogenesis, which again you heard about from Sarah Fortune just now, optimizing cost effectiveness, which Peter White showed you the principles, but we need to apply those principles to the case of whole genome sequencing over the next few years.